White skin has been the main racist issue. There are white colored people in many parts of India, particularly namely Kalash, Nuristan, etc. A white individual does not carry a black skin color gene. On the other hand, a brown or black individual carries as recessive within it white color gene. According to the evolutionary theory, the original population has always more variations, and purified populations or pure races originate out of this population by a process of isolation. This means that the white population of the world could have originated from the darker people by eliminating the black color genes from them. That's why the white color gene is widespread in India, even in South Africa, although out of the two alleles, one is dark, therefore the person looks dark. So it is by the process of elimination of the dark color gene that white color race evolved. Haag 2015 and Olalde 2014 found that the people of Europe had dark skin color up to 5000 BC. They acquired the white color from the Asian migration migrating to Europe after 5000 BC. The Indians' migration eliminated the original European population to extinction in most parts of Europe, leaving a few percent of them in the southwest Europe only. Ellen Toft in 2015 found that Europeans acquired the light skin color between 5000 BC and 3000 BC from Asia. There are so many light colored genes in the world. Some specific to China, some specific to Southeast Asia, and some other for Amerindians. The Indian light skin color gene is exactly the same as the European one. This map shows that the green areas are the places where population is pure white. This distribution of a particular light color gene from India to Europe through Iran prompted Basu Malik, Ganeshwar Chaubey, etc. to claim in an article that this migration had taken place from Europe to India because the genes were identical. However, Keith Cheng 2014 proved that the reverse was true. He found that the light skin in Europeans stems from one 10,000-year-old ancestor who lived between India and the Middle East. The pattern of people with a lighter skin color mutation suggests that the A111T mutation occurred somewhere between the Middle East and the Indian subcontinent. This mutated segment of DNA was itself created from a combination of two other mutated segments commonly found in Eastern Asians. The combining of the segments occurred after the ancestors of East Asians and Europeans split geographically. One thing is important to understand that in current vocabulary of the geneticists, the Middle East includes Pakistan and Afghanistan along with Iran, and the West Asia includes Iran but excludes Pakistan. Now, the combining of segments occurred after the ancestors of East Asians and Europeans split geographically. Now, this postulated split had actually taken place in southern India as deciphered by the IBM study of the recombinant autosomal DNAs. So soon after the split, possibly the two genes which are independently found in the eastern Asia got united in South India to give birth to the white color gene. So, from the same set of data, a more scientific hypothesis would be the split between the eastern and western DNAs took place in India as depicted in IBM map. The light color gene originated in South India after this split. It did not undergo natural selection in South India because light color is harmful in South Indian location, causes skin cancer. The strong sunlight say at the onset of the Holocene, migration in India took place from south to north. Undergo natural selection in South India because light color is harmful for the people of South India because it causes skin cancer. The strong sunlight provided adequate vitamin D even to the dark skinned people in South India. Hence, the light skin had no survival advantage over the dark in South. Its frequency remained low. Some people with the light skin gene came to North India. Some settled in the Himalayas, Kashmir, Pamir, Kalash, Nuristan, Himachal, etc. They received poor sunlight, particularly due to heavily covered body to protect from the cold. They became vitamin D deficient and became inferior in the struggle for existence. Light skin color gene was selected by natural selection in this golden age. Now, another such story is about milk digesting enzyme lactase persistence. Not everyone in the world can digest milk in the adult life. However, people of some parts of the world can do that. This is because the lactase persistence gene. There are several different LP gene or lactase persistence gene in the different regions of the world. South India has lesser frequency of the LP gene, yet there are more than one varieties of them. Now, the finding of the commonality of the LP gene of India and that of Europe prompted Romero, Ganeshwar Chaubey, Kiwi Seal, etc. to claim that the farming, domestication of cow, and drinking milk started in Central Europe in 5500 BC, and then these people migrated to India, bringing farming and milk drinking within next 500 years by the time 5000 BC. And they conclude the earliest evidence of cattle herding in South Asia comes from the Indus River Valley site of Mehergarh and is dated to 7000 YBP, years before present. Meadow 1993, thus post-dating the predicted start of the European Neolithic sweep 7500 YBP. It is therefore plausible that from Europe the allele subsequently spread into Central Asia, the nearest Pakistan and India, although not necessarily by the same route or the same demographic event. But actually, Medo had written 7000 BC. Thus, from converting the date from BC to YBP, years before present, they manipulated 2000 years, and this gave the impression that the migration, origin of farming, and pastoralism originated in Europe earlier by 500 years than India. However, the fact is entirely different. In Europe, pastoralism did not occur till quite late. However, other studies indicated India is the source of the European LPG. No LPG in Europe found before 1000 BC. in the work by Olalde and Lalueza Fox. They wrote, the conspicuous absence of the persistence allele in many Neolithic individuals from different places in Europe. In a recent study of 5 millennium transect from the early Neolithic to the Iron Age in Hungary, and the first LP allele appears in a sample that is only 3,000 years old, suggests that the rise of LP mutation could be related to pastoralism practices spreading during the late Neolithic or even later. Apart from the light skin color gene and LP gene, is the Indian domestic cow had migrated to Ukraine, Mongolia, China, Africa, and other regions. A Food and Agriculture Organization picture shows that Indian cows entered Africa through two routes. One is the northern Sinai route, and that they followed the upper border of Africa to reach the West Africa, and this population carries the Indian variety of LPG. Then there were other people who, between 3000 BC and 1000 BC, entered through the Horn of East Africa through the sea route with cows, and they spread Indian cows in Africa, that region of Africa. So the cows are of Indian origin there, but the LP gene, they had another LP gene evolved within them, and so LP gene are of two types. Chen in 2010 and Catalan in 2009 studied the DNAs of different cows in Asia and Europe and also Africa, and they found that the humped cow all over the world have originated from Europe. This means that the milk digesting gene reached Europe from India as late as late Bronze Age. Another type of evidence could be the mice were adapted to agricultural life in India, and they migrated with the Indians to everywhere, wherever the Indians went during the Neolithic migration. Now, the migration route maps have been drawn accurately on the basis of the genetic distribution of the mice in the world, and they all indicate that from northwest India, two routes were adopted. One was the southern route along the southern coast of Iran, going into North Africa and then South Europe, whereas there was another route eh, which people, which mice traveled along Central Asia and reached Caucasus and North Black Sea and then Northern and Central Europe. Then there is another evidence of scientific evidence of Indian migration to Europe. That is Otzi man, 3300 BC, Alps. A frozen mummy of Otzi man was found who had a stomach infection. Bacteria was isolated and found to be H. pylori. And this H. pylori sample was exactly the same which infects in India. That means people with this infection had migrated there from India. Now I am coming to the last point, and the time being short, is R1A controversy. R1A has been identified as a marker of the Aryan or Indo European migration. Now it is distributed in India, Middle East, and Europe. Underhill in 2010 wrote, importantly, the virtual absence of M458 chromosomes outside Europe speaks against substantial patrilineal gene flow from East Europe to Asia, including to India, at least since the mid Holocene. What he means to say is that Although the Indian branches of this particular lineage are found in Europe, but European branches they are not at all found in India. That means no migration took place from Europe to India. However, Underhill had to suffer a lot. However, this was not appreciated by the Eurocentric researchers. Underhill in 2015 did research over R1A branches again, and he found that the proliferation of the branches, the initial proliferation of R1A, did occur somewhere in the vicinity of Iran and not in the steppe or in Europe. We can say at the onset of the Holocene, migration in India took place from south to north. Some people with a light skin gene came to North India. Some settled in the Himalayas, Kashmir, Pamir, Kalash, Nuristan, Himachal, etc. They received poor sunlight, particularly due to heavily covered body to protect from the cold. They became vitamin D deficient and became inferior in the struggle for existence. Light skin color gene was selected by natural selection. You can see Karelian hunter, a bone has been found from Karelian region 3500 BC. Now, one interesting finding is that Indian dog migrated with R1A people. Cherry tree cave CTC dog found a skeleton found from Germany, which dates to about 2800 BC, was found to have Afghanistan type of dog DNA. R1A distribution map shows that Karelian hunter of 3500 BC was the oldest sample recovered from Europe. And then other samples are about 2500 BC or later. So there is a correlation that humans migrated to North Europe not only with R1A DNA but also with Indian or Afghanistani dog, village dog. Now we come to see how the branching of R1A has taken place. This map depicts the distribution of the M17, which includes all its descendants, and we find that it is widespread from Europe, Siberia, Central Asia, Mongolia to India. And of course, it spreads to South India also, which hasn't been shown clearly in this picture. But this picture certainly shows that this lineage extends to Southeast Asia, Malaysia, Asia, Madagascar, Central America, and South America also. We are aware that there were great navigators in South India who could have gone through sea and settled in Southeast Asia, as well as they would have migrated to Central America. R1AZ645 gives two branches in northern Indus Valley region. One is Z283 and the other is Z93. Z93 is distributed from the Himalaya region of India up to Mongolia and uh, Siberia, whereas the 283 goes to Central Asia and then spreads to Mongolia, Siberia, Western Siberia, and however, we can see that the Z283 does not try to come to India or Iran, implying that there was no migration from Central Asia to India or Iran, although from Central Asia people migrated to Europe, towards the east to Mongolia and in the Siberia. Its European branch of 283 is 282. So we will see how 282 behaved. 282 became divided into further branches, which are further distributed in Europe. None of them turns towards either Central Asia or India or Middle East. They are European and stay in Europe, indicating that no migration took place from Europe to India. Otherwise, some of these individuals would have come with that. South Asian branch of R1A is a Z645 is Z93. Now, Z93 has two main hotspots. One is Himalayan Kashmir, Himachal Pradesh, North Pakistan area, and the second is Mongolia. Now, Tarim Basin mummies dated 2000 BC. A large number of mummies have been found from Tarim Basin in Xinjiang, and they are all white colored and they contain mitochondrial DNAs M5 and Indo-Iranian DNA U7. Tarim mummies also have the Iranian mitochondrial DNA T2. Besides this, Indian male lineages R1 M173, R1A1A, R1 M417, etc. Senior lineages were also found. However, most dominating was M93.